Yeah, well, thank you. And hello, everyone. I'm now going to talk about the use of metabarcoding for the characterization of freshwater nematode communities. And I will present two of my studies, which are part of my PhD. So starting with a brief introduction, although most of you already know all this. So nematodes, of course, are one of the most abundant groups in the meiobenthos, with about 80% of all multicellular organisms. And they exhibit a very high diversity. They have a lot of different feeding types and, of course, a high species diversity with about 28,000 species, while other estimates suggest about 1 million species. They are also key organisms in the pelagic food web. So they feed on bacteria and fungi and small protozoa, and they are the food for macrofauna and small vertebrates, such as juvenile fish, for example. They are furthermore suited bioindicators. So they are typical species for unpolluted sites, and in contrast to macroinvertebrates that are usually used for bioindication, they are also present in the whole year. So the identification of nematodes for about the last 150 years, it had been carried out by morphology, so using specific characteristic morphological features to distinguish between species. So for example, looking at the mouth area to distinguish the species, but as you can see here, those features can also be very similar. So there's a lot of taxonomic expertise needed and a lot of training to reliably distinguish between the species. So besides the morphological identification, there's now the method to use molecular species identification. So in general, there are differences in the DNA that are used as the basis for the identification. So if species A and B are morphologically different, they are also different in their DNA structure. So those genetic sequences can then be used to be stored in a database. So if I have an unknown specimen with an unknown sequence, I can compare those unknown sequences to my known sequences in the database. And if there's a rather low accordance where I can't identify my specimen, but if I have an identical or very, very similar sequence, I can identify my specimen. So in the last decade, there have been many, many projects with the aim to collect the global biodiversity. So genetic databases are quite equipped for some taxa, at least. So, one method for molecular species identification is Sanger sequencing. So basically there's one specimen which is extracted from the sample, then there's a DNA extraction and a PCR with specific primer pairs, and Sanger sequencing, of course, and in the end we have one specific sequence for our individual. Then since the last decade or so, there's also the option to use next generation sequencing now. So the main difference to Sanger sequencing is that there are several thousand sequences generated in one single run. So compared to Sanger sequencing, it's a huge time saving, and it's also more cost effective. And I'm now going to talk about metabarcoding, which uses next generation sequencing and amplicon sequencing. So using specific short gene fragments instead of a whole genome, for example. So we can use now bulk samples. So we can use all our myofaunal group, for example, and use it as a bulk sample and sequences it. So what molecular studies have now been conducted with nematodes so far? While in general, the marine and terrestrial species have been well studied. So the plant parasites in general, because they also have an economical impact. So for nematodes or most eukaryotes in general, the gene fragments that were amplified the most are ribosomal genes, for example, the 18S gene fragment and the 28S gene fragment. And of course, there's the mitochondrial DNA, and for example, the CO1 gene region, which was also initially the barcoding region of choice. So for freshwater species, there have been very, very few studies so far. One of the first studies was with Tuberous gracilis. As we can see here, we could show, or it could be shown, that there's a high genetic diversity in this species. And most studies so far used Sanger sequencing, but there of course have been some NGS studies with the focus on nematodes, for example, with marine species or soil nematodes. So I'm now going to talk about the quantification of metabarcoding. So the quantification of metabarcoding has not yet been approved so far, although quantification is needed if abundance-based data is used. So the number of reads is generally influenced by biological factors such as the gene copy numbers, or technical factors like a differential amplification efficiency between species. And a lot of studies suggest that the biomass is one of the most impacting factors for the quantification, even more important than the abundance of these specimens. So we conducted a metabarcoding experiment with the artificial nematode community, so a mock community of known abundance and biomass. For that, we used 18 different species that were cultivated in our lab, so it were very clean species. They were extracted from the plates and combined into new communities of 200 specimens each. And previously, they were photographed and measured, so the biomass could be calculated later on. So each community had 200 nematodes, and there were 3 replicates for each proportion that was used. So we had 9 different proportions that we see here. We had, just very short, we had proportions with an equally distributed species and specimen number, and we had rather natural proportions, for example, with, a very dominant species and the others at lower proportions. So for the metabarcoding, we used a 28S gene fragment as marker and 2 18S gene fragments. And the hypotheses were that the diversity would be detected. So basically all the 18 species should be discovered, and that the biomass would be a more important factor regarding the quantification than the abundance of our specimens. So here's the first result. We have an NMDS plot with our 9 different communities. In bright blue, there's the original or true community regarding the abundance, and the black points are the communities in regard of the biomass. So in green now, we have the genetic data. We have the 18S V1 region. So we called this marker 18S V1. And we can see that basically all data points cluster in this area. So there is no overlap to the community, neither in regard of biomass or abundance. But if we look at our second genetic marker, which encodes for the 18SV4 region, so 18SV4 in darker blue, we can see that there's indeed a slight overlap to the black data points, so the true community in regard of biomass. We see our 3 replicates from the genetic data, for example, for the S4 region, and it's quite close to the data point for the biological community, for the community in regard of biomass. So this pattern becomes even more clear if we look at our third genetic marker, which was the 28S gene region. And we can see that the starter points in red, for example, the S2 community, the 3 genetic replicates are very close, and also the data point for the biomass community is very, very similar. And this pattern can be seen for almost all communities here. So there's a great similarity between the biomass proportion communities and the genetic marker. So this in a correlation, on the x-axis we have the relative biomass, and on the y-axis we have the relative reads. On the left side for the 28S marker, which was the formerly red one, and we can see that we have a very high correlation with an R-square of over 0.9. And for the 18S34 marker, which was the blue one, we had still a good correlation with an R-square of 0.8. So overall, this was more distinct for the 28S marker, as also as we could also see in the NMDS plots. And third genetic marker, the formerly green one, did not yield any correlation, so it's not seen here. If we now compare it to data of relative abundances, so the community of relative abundance on the x-axis and relative reads on the y-axis again, we basically have the same pattern, but with overall a lot weaker correlations. We have for the 28S gene fragment an R-square of 0.5, and for the 18S34 region an R-square of 0.3. So we can say that the biomass is definitely a more important factor regarding our quantification ability. If we now look at the diversity that we could discover, from our 18 species, up to 16 were discovered. So we had one species, this one here, that could not be discovered by any of the genetic markers. So there was a Sanger sequence that could be generated. Yeah, so there are no mutations in the primer binding region that prevent an amplification in general. And furthermore, there were some species that could not be differentiated at the species level. So for example, these 3 Plectus species could not be differentiated, so they always had the same sequence, with the exception of Plectus velox for the 28S gene region that could be differentiated from the other 2 Plectid species. So this is a known problem because the ribosomal genes tend to evolve rather slow. So it's a trade-off between the taxonomic resolution and broad amplification efficiency. So furthermore, the 2 species here, Panagrolimus tinemani and Diploscapta coronatus, both have the lowest biomass that they contributed to the community, and they also had the lowest amplification success. So we can say that there's a tendency that species with a high biomass impact tend to be found rather easily in the amplification. So as a conclusion of this study, where the majority of our species could be detected and some species were not amplifiable or differentiable, that's caused by the slower evolutionary rate of the ribosomal genes. And the primer efficiency is also not equally distributed. So a mutation in the primer binding region can already cause a difference in the amplification success. So that's of course not equally distributed between the species used. The quantification, well, at least with our artificial community, was indeed possible, at least for 2 of our markers. And as hypothesized, the relative biomass was in a good correlation to the relative Reed numbers. So the biomass is indeed a more important factor than the abundance. This is also consistent with other studies. For example, on ciliates, they had similar results. So in the second part, I'm now going to talk about the molecular bioindication. And so far, molecular bioindication using next generation sequencing had been successfully shown for marine macroinvertebrates and also for diatoms. So there's strong potential of NGS-based data, but so far there were no molecular-based bioindication studies with freshwater nematodes. So we conducted a study for the molecular assessment of sediments using the Nemesis Index. And for those of you who haven't heard from the Nemesis Index, this index has demonstrated that the sediment quality can be depicted by nematode communities. So nematode species are classified into species at risk and species not at risk, just like for the spear index for macrofauna. For example, the species at risk are typical for very unpolluted sites. The Nemesis Index is then calculated by using the relative abundance of those species at risk and the relative abundance of all species found. And the index showed that it can classify the sediment in regard of the pollution into different classes. So we have here the pollution and here the Nemosphere. And on the left side, we have the species level, and on the right side, the genus level. And we can use these classes here: bad, poor, moderate, good, and high, just like for the spear index again. And yes. So that was already known so far, and we now validated this index with molecular data. So for that, we used 7 different locations with a varying sediment quality that was known from further previous studies. So these are the 7 locations across Germany. For the metabarcoding, we used 18S gene fragment and a 28S gene fragment. So the 2 genetic markers that performed well in the quantification approach. We calculated the Nemosphere based on morphological data and based on molecular data. And we did this for the species and the genus level. We then compared those data. So the hypotheses were then that the nematode communities of the different locations would be different according to their pollution status, and that the Nemesis Index could also be used with molecular data, for example, that the same classes should be achieved. So we now have here the results of the Nemosphere index. We have a pollution gradient here. So as shown here by the mean per Q value, we basically have 3 sites that were very, very unpolluted. We had 3 sites here that were moderate polluted, and we had 1 site that was very polluted. So if we now look at the Nemosphere for the morphological data, so the traditional Nemosphere, we can see that the values basically follow the pollution gradient. We have the highest values here and the lowest value for the most polluted location. If we now look at the genetic data here for the 28S gene region, we have basically the same trend with the exception of the location RM. So the value is very low there compared to the morphological data, and it's also not fitting to the gradient. For the 18S data, we have these 3 locations classified as not so good or even poor, and this one here again as moderate, so it's a little bit shaky. So on the genus level now, we have for the morphological data, we have again the same gradient, but the overall values are a little bit higher than for the species level. For the 28S gene region, we again have the same trend, and again we have the location RM classified as way lower than for the morphological data, and the 18S data for the genus level tends to follow the gradient quite okay. So again, we have the 28S marker with a slightly better depiction than the 18S marker, and we can say that the genus level shows a better concordance than the species level. So this again in correlation. On the x-axis, we have the Nemesis based on the morphological data, and on the y-axis based on molecular data. On the left side here for the 28S gene region, we have a correlation with an R-square of 0.4. So we basically have these two data points here that prevent good correlation. So at this side here, the Nemesis based on morphological data is quite high, but on the molecular data, it's not so high. But that can be reasonably explained because the two species, Eumenhystera vulgaris and Eumenhystera longicordatula, both make up over 35% in the morphological determination. And for both species, there is no 28S sequence available, so we can't find it. The same is found at the RM location. We have the two species Punctodora ratzepogensis and Monhestrella paramacula, and again, both are missing a 28S sequence. So the rather weak correlation here can reasonably be explained. For the 18S data, we could not find any correlation at the species level. If we now look at the genus level, we find a very good correlation for both markers. We have again the morphological data on the x-axis and the molecular data on the y-axis. For the 28S gene fragment on the left side here, we have a very good correlation with an R-square of over 0.8. And for the 18S gene fragment, we also have an R-square of 0.7, where the overall slope is a little bit weaker. So the values for the genetic data tend to be very lower than the values of the morphological data. If we now look at our different nematode communities, we have here an NMDS plot with the genus level data. So because it's obviously showing a better depiction, we have our location depicted by the method of identification. So black points are for morphological identification. The white ones are for the 28S genetic marker, and the gray squares for the 18S genetic marker. And again, we see that the rather unpolluted sites here cluster on the left side of the plot, while the more polluted sites are here. And these two locations here are a little bit on the right corner, a little bit separated, but they both belong to the same river system, the Elbe, so it's quite reasonably that they are very similar. The dotted circles indicate a 20% similarity by a simple analysis. So we can say that the method of identification is not important, at least for these data points, because the communities are very, very similar, while the exception is these two data points, which have significantly different communities regarding the identification method. So as a conclusion for this study, we can say that the locations can indeed be differentiated according to the nematode communities, and the factor location is more important than the factor method, at least for most of the locations. And the Nemesis Index is also applicable with molecular data, especially at the genus level. So that can also be explained if we look at the species of risk. So there are 58 species of risk for nematodes, and only 38 of them have 28S sequence at the moment. So on the contrary, 62% of the species are still missing a sequence. And if we look at the genus level, over 70% have a sequence. For the 18S gene, where 55% of the species at risk have a sequence, and even over 90% of genera. Well, of course, we only used a very small data set with 7 locations, and to really say that the Nemesis Index can be used for bioindication, of course, larger data set is needed and a further validation, but it's a good point to start from. So general conclusions for my PhD are, well, there's a general applicability of the metabarcoding for the characterization of freshwater nematode communities. And species identification, well, especially at the species level, is often very difficult due to the database shortcomings. The quantification, at least with the ribosomal gene fragments and the specific primer pairs that we used, is also possible. Well, of course, the use of presence-absence data to avoid a quantification in general is also suited, but for example, for bioindication, often relative abundances are needed. So, as I already said, the molecular bioindication is also possible. So this might allow to circumvent the taxonomic deficit that is known for nematodes. Well, for freshwater nematodes, I think there's currently only one expert that's able to identify them. So they might be of broader use if the molecular bioindication can be used. Well, of course, further efforts for the database curation are certainly helpful to increase the taxonomic resolution at the species level. Yeah. So, well, I'd like to thank, of course, my working group, which is the animal ecology of Bielefeld University and the Federal Institute of Hydrology that funded my PhD. So I'm happy to take any questions.